structure is made, the structure, this is the so-called primary, the protein, it's made as one long strand, and then it folds, F-O-L-D, it folds. It folds to the point, and you don't have to know what all of these mean. If you were seniors, you would, but you don't. It folds upon itself. It folds, F-O-L-D-S, until it has shape, a specific shape. And that shape is by all of these interactions. Hydrogen, here's just to show you, this is a hydrogen bond interaction right here, subhydro bond interaction right here, ionic bond interaction right here. That's because you don't have to know this, but they're folded based on attraction, positive, negative. Like for instance, if I have a positive right here and a negative right here, it's going to start the folding process. Okay? I want you to know about this procedure. I want you to know what the molecules called chaperones are. It keeps going back to chaperones. It keeps going, reverting, the default is that big thick. Okay? I want you to know what chaperones are. Chaperones are proteins that make sure that the other proteins are folded properly. Once they are generated, once the protein is made, the chaperones are going to make sure that they are folded properly. Because if they are not folded properly, once they come out of that cell, they're not going to work well. So they have to be shaped perfectly. Okay? And that's what chaperones are. Okay, now let's go on to let's see, I'm going to skip over the motif, you don't have to know that. Here's what the chaperones are. The chaperones make sure that everything is folded correctly. It has to be folded correctly. Okay? Folded correctly. That's their function. They make sure that it's folded. If it's not, the protein will not work. The proteins in your body they have a certain structure. In class I use the term, it's like pieces of a puzzle fitting together. One piece interacting with the other piece. Okay, all right, now I do want you to know this term denaturation. Denaturation is the process where you break all these bonds. You pull it apart. I think the example here's the folded, this one is folded, and here is the unfolded. And you're breaking all those bonds, and it becomes unraveled. Think about it as being unraveled. I mentioned in class that remember the example I used of boiling water? I asked you why you boil water after there's a natural disaster. You boil water because you're killing microbes, you're killing the microorganisms, and you're killing them by unfolding their proteins. If you unfold their proteins, they will not work properly. So you boil water after a natural disaster to kill microbes. And during that process of killing the microbes, you're destroying their protein. You're destroying the protein structure that go from a folded molecule to an unfolded, and they do not work like this. Proteins don't work in this structure. Are you with me? So the boil is going to heat everything up, and heat has, according to the physicists, it expands, and it's expanding it from here to here. Are y'all with me on that? So that's what denaturation will do. It will lose the structure. Okay? It's going to lose the structure. Lose the structure, and then it's not going to be able to work. Meaning the function is also lost. Because the protein, the protein has to be it has to have a certain structure. If the structure is eliminated, it will not work. It won't work. Okay? And denaturation will destroy And actually, here's what denaturation will really do. Denaturation will, if I go back to my protein structure, it'll bring back, all you have is this, it'll just be one long strand. You won't have something that looks like this, where it has structure, where it has folds. Do you see the point? The difference between a long strand and this right here. Okay? Are you with me on that? For protein, yes or no?
Yes. Okay, if you could erase some of the coloring, I can't really see it.
Okay. Well,
That's really
well,
because it just follows.
Yeah,
it's
fold.
different. Okay.
that's all I need. You don't need to see the specific kind of folding. If you were a senior, I would ask you that. So hydrogen bonding interactions, covalently, every other, but the point is, without folding, it's gonna look like this, just a straight line. And with folding, it's gonna look folded. And you're unraveling it. Think about that. When you heat it, you unravel it, and that destroys it. Okay? That's why you boil the water. You're actually unraveling those bacteria, and you're sterilizing it by doing this, because it no longer has the protein, and actually denaturation also is related to destroying the DNA. Those individual strands of the DNA, they become separated. And anytime you take away separate the strands, you're gonna destroy the DNA, right? So that's what boiling will do. That's what boiling will do. Okay, so let's go to the lipids. Same dehydration. Remember that lipids, everything in the lipids, hydrophobic. It's cholesterol, estrogen, testosterone, those are also hydrophobic. Fats, everything that hates water, hydrophobic, is going to be in this thing. Okay, there might be a difference. I'm definitely going to ask you about a phospholipid membrane. All membranes are made up of phospholipid. All of your membranes are made up of phospholipid. It's a phosphate group and a lipid group. Okay? All of your membranes are made up of phospholipid. Okay? And they call it a bilayer, but I don't necessarily think you should know that, but I want you to know, I might just say a phospholipid bilayer, but it's got to have that term phospholipid in it. All membranes, all of the membranes, which are the surfaces around your cell, the surfaces around your nucleus, the surfaces around your mitochondria. Do you see the point? All of the membranes in your body are made up of phospholipid. All of them. All of them. So this is an important structure. And here's the phosphate portion right here. And here's the lipid portion right here. Saturated, unsaturated fats. Okay. And I talked about that. And I said that, depends upon what you eat, whether or not you eat potato chips or avocado. Good fat is avocado, potato chips are bad, generally. Okay, so anyway, this just shows like this would be like the outside of the cell, which is mostly a water environment, the inside of the cell, which is water environment, and here's the surface of the membrane, the surface of the cell, and the surface of all of your organelles, your mitochondria, the surface of all of your mitochondria, surface of all your nuclei, the nucleus, all of you are made up of phospholipid. Okay, are we good? That's the end. Okay, now you had asked about chapter 1. I'm going to go over chapter 1 so fast your head's going to spin, they say. Okay, because I really do have to go at 4 because I'm running an experiment in my lab. I want you to know what defines life. Okay? I think all you have to do is compare yourself to a rock, it's pretty easy. I'm not gonna go through everything that's in this chapter, I'm gonna skip over a whole lot. I'm skipping over all of this scientific reasoning, all of this. And then I'm going on